Every year, I am asked to give a presentation about the World Vegetable Center breeding pipelines. This presentation usually overlaps with the presentations of the breeders because they talk about their breeding pipelines. So next slide, please. So I take the freedom to just touch the breeding pipelines very, very quickly, and then tell you other news of the World Vegetable Center. Here, this is a very similar slide to the one that I showed last year. This describes very quickly the products that are in the pipeline. Next slide, please. And the major developments, of course, is virus resistance for all our crops. Now we give more emphasis again on bacterial wilt, especially on tomato, but also on pepper. And then foliar diseases are gaining importance. We have seen that Alternaria is becoming a big problem. Early blight is becoming a big problem. And of course, we are working on nutritional traits, on quality traits, fruit size of tomato, pungency of pepper. But more details about the breeding pipelines you are getting this morning and in the early afternoon from our breeders. Next slide, please. So I would rather like to profit from my 10 minutes to tell you other news about the World Vegetable Center. Last time I already showed you the high throughput phenotyping facility that we have built up at our center. And in the last 3 years, we have done experiments on pepper, amaranth, and okra core collections. These core collections are representing the diversity that is present in larger germplasm collections. And the aim of these experiments was mainly to identify heat stress tolerant pepper materials for breeding and to identify flooding tolerant okra accessions, also for breeding. In Derek's presentation, he will show us some of the results of the heat stress tolerance trials using this high throughput phenotyping facility that were performed over the last 3 seasons. Just very quickly, this high throughput phenotyping facility is not taking pictures of plants. It is monitoring the growth of the plants by generating a 3D data cloud of each individual plant. And this 3D data cloud then can be easily analyzed by an appropriate software to get in an automatical manner morphological and partly also physiological data of the plants. We get about 3 measurements every day. So we can produce quite exact growth curves over time, and we can really see how the plants behave. I call this machine a machine that helps me to watch the plants growing. Next slide, please. The data then can be analyzed in multiple fashions. You can take the data directly and you can look at the growth curves, or you can, as it was done by my colleague Lin Yaping, to use some software that allows you to do machine learning or artificial intelligence approaches to extract more information from this data. So again, for example, from the heat stress trial on pepper over 3 seasons, we were interested in which trait changes at which temperature most. So we saw that, for example, NDVI, this is normalized differential vegetation index, an index that shows plant health, and a senescence index in average changes most at the temperature around 30 degrees. With this information, we can design better experiments at different locations or in different seasons to make observations on our germplasm. Of course, most interested we were in the variation of traits under heat in the germplasm panel. And also here, machine learning approaches helped us to identify materials that are more tolerant to heat than others. Next slide, please. On this a little bit complicated slide, for example, I try to show you a principal component analysis for all traits, and I show the vectors for some traits. The vectors that show upwards, these are traits like plant health. These are some punctual values. The vectors that show downwards are those traits that accumulate over time. This is like a leaf area, biomass, these kind of things. And then you see the points in the PCA. The points in the PCA, these are the entries. These are the genotypes that were tested. Those that are to the right of the center are those that show a greater reaction under heat stress, and those to the left are rather candidates for heat stress tolerance. These are those that react less under heat. And like that, we can rapidly identify accessions that are more useful for breeding than others. The advantage here is that we are not looking at one trait at a time. We are looking at a larger number of traits, and we see the more complex reaction towards heat than if you would look just at biomass or these kind of traits. Next slide, please. And another novelty at the center is that we are planning to introduce a project that allows us to build up a system for systematic monitoring of pathogen populations by using a molecular marker. In the audience, you can meet Ricardo Oliva. This is our pathologist. He is coming from the International Rice Research Institute, where they have already established apparently a similar system to monitor rice pathogens. Next slide, please. What we are hoping to do in the next few years is to build up a monitoring system for major vegetable crops in major vegetable growing regions as an early warning system and also as a decision support system for managing diseases. And we are about to starting this initiative, and I hope that next year at the workshop, Ricardo can already present how this system is advancing and how we are using it for monitoring diseases, these species, pathogen species, and even pathogen strains across regions. So next slide, please. The research focus for our group for the next year is, of course, still phenotyping, using this high throughput phenotyping device that we are running now at our center. By doing this, we are adding information to our germplasm. We are then not just distributing seed, we are distributing seed with known phenotypes, with known traits. And that, I think, adds enormous value to our germplasm collection. And this kind of tool also is supporting breeding by identifying source materials, of course. And we get a much better insight into trait and trait diversity of our material by using this phenotyping devices. And of course, once we have phenotypic information, and genotypic information nowadays is very easy to generate, we can combine these two kinds of data for producing molecular markers that help the breeders to do marker-assisted selection in their program. And when it comes to breeding, of course, we are continuing to develop traits for the breeding programs and to produce high-performance lines for direct release and hybrid development. Here in breeding, not much has changed. Breeding, as everybody of us knows, is a long-term effort, and we are not changing our research focus now in breeding for 2022 and 2023. Next slide, please. So I came to the end, apparently, and I wanted to thank you very much for your attention. And we have still a few minutes time for questions and comments to this part of the workshop. Thank you very much. I can see a question online. I will read it out now. The online question is coming from Balram, and it seems that this is a colleague from AVBC, from the African Vegetable Breeding Consortium. And the comment is pathogen diversity studies. Would that be done at Hyderabad, at the Hyderabad Center? For the samples for India, or at the World Vegetable Center? Oh, I'm corrected. It is not AVBC. It's very small to read for me. I think I need glasses. The question is coming from Nunhems, of course. It has to be an APSA member, APSA-WorldVeg Consortium member, to be able to participate with this meeting. So the pathogen diversity studies, as far as I understand, are coming directly from the field across the region. So this is not done at a certain center. The pathogens are monitored in the growing regions, and this information is fed in an information system and is then provided to stakeholders. But please, may I ask Ricardo to answer this question in a more correct way than I could do it?
Yeah, I mean, you are right completely. And the only caveat there is that it will depend on the pathogen, right? Each pathogen will have its own strategy for detection monitoring. But in a broad aspect, what we want is centralize that information so everybody can see the same thing. And then there is when we start to have regional strategies to control pathogen, and that is when things start to change, hopefully. So in terms of detection, it will happen here or there. We still have no much decision on that, but definitely what we want is to develop molecular tools that facilitates the monitoring in the field and fast track the information to the final users, which obviously have to be breeders or extension service, and so many stakeholders that depend on that information.
Thank you very much. There seems to be, yes, there is one more question in the fifth row.
Yeah, this is with respect to the pathogen diversity studies. If it is done centrally, how do you address the quarantine system which prevents the import of pathogens?
This question was coming from the virologist of IIHR. Sorry, I did not get your name.
Dr. Krishna Reddy from Head Division of Crop Protection, IIHR, Bangalore.
Mm-hmm. So as far as I understand, the pathogen identification is done on site, but I hand over again to Ricardo, who can answer this question much better than I could.
Yeah, again, so for instance, I will put one example. We are working on bacteria wilt or bacterial leaf spot pathogens for bacteria, right? And we are using genomics to try to characterize the population worldwide. With that information, we want to develop simple SNP markers that can be run here in Hyderabad, or by even not necessarily the sample need to go to Taiwan or anywhere, just can be genotyped here, very in a matter of days, and then the information can be uploaded. And that won't entitle any pathogen as the sample as a DNA that can be moved. So that's some sort of the strategy. And again, it depends on pathogens. As you know, viruses may have a different context. They have RNA virus, DNA virus. So that's a different context. So we need to take case by case decisions, a collection of pathogens in general.
And also our colleague, Dr. Balaram from Nunhems in in the chat box commented on that. He suggested that the molecular studies can be done on samples that are collected from hotspot locations. And I suppose that it is not a big difficulty to send then DNA of this material around to the next laboratory that can work with that. So for this time, I do not see any more questions and comments for this part of the vegetable breeding update. Before we continue, let's break for coffee, and the coffee will be served in the IIHR food court. This is in the back of this audience. I just see that there is one more question coming in online. If I may have another minute before the coffee break. The question is coming from Dr. Prasad, and he refers to the 3 years of high throughput genotyping of pepper, amaranth, and okra for heat and flooding study. What was the findings? Well, we're talking here about high throughput phenotyping of pepper, amaranth, and okra. What was the finding? For okra, we found some accessions that are more tolerant to flooding, that continue to grow during flooding, and that continue to yield also during flooding. And the results of these trials were published. And for pepper, the result is of course more complex because this was done over 3 years, and the principal findings will be presented a little bit later by Derek. But overall, the finding was that there is variation present for heat stress tolerance traits. There are some tolerance traits that are known, other traits are new. Derek will provide the surprise about the new traits. We found the interaction between certain traits and correlations between traits, some of them obvious, others surprising. Briefly, these kind of experiments allow us to select germplasm as source material for heat stress tolerance breeding in a much more rational way than it was possible before. And it's also possible to find traits in larger collections. So this automatic phenotyping in the field is relatively cheap. So it allows to look at germplasm in a much more careful and diligent way than it was possible before. I hope I could answer this question from Rallis India, for the moment at least appropriately, and I trust that Derek will shed more light on this. So it seems now it's time for us for having a coffee, for breaking for coffee. And please let us be back again a little bit before 11:40 to continue the session. Thank you very much.